Resume our discussion. Did I get louder here? Or? Sorry. Okay. Are there any questions from the first hour? Okay. Believe we left off with our discussion of the strategy for diagnosing a vitamin deficiency. Now, not shown here, and that should maybe be a 2.5 bullet point, is measure the vitamins in the serum. However, that's not always useful because for some vitamins, the amount that's circulating in your blood is going to be maintained at within a pretty tight range, even if your stores of it are depleted and the functional amount of it is low. Okay, so for vitamins, looking at how much is in your blood is not always a good way to diagnose the deficiency. Okay, so with the erythrocyte glutathione reductase assay, the idea is that you take an enzyme that requires the vitamin that you're interested in this case, the vitamin riboflavin and the cofactor FAD, and you look at its activity in a sample that's easy to obtain from the patient. Okay, so you take a patient's blood cells, you lyse them, and then in an in vitro assay, you set this situation up. You have oxidized glutathione and then a bunch of NADPH, and then you let the reaction go and you look for, NADPH. You can look for that spectrophotometrically, so over time the NADPH will go down. Okay? And you can measure the amount of activity of that enzyme or the rate of NADP plus production and plot it. So if we have two patients here, and this is their erythrocyte glutathione reductase activity amounts, can we tell which patient is deficient? Patient 2? What if patient 2 just has a erythrocyte glutathione enzyme that has an intrinsically lower activity? Okay, so what you do here, the coefficient is referring to this reaction both in the absence and in the presence of a ton of extra FAD. Okay, so if you add a lot of extra FAD and you don't get a big increase in activity, the interpretation is that that enzyme is fine. It's got plenty of the cofactor it needs. If you add extra FAD to the in vitro reaction and you get a big increase of activity, the interpretation is that the enzymes are starved of FAD in that patient. Does that make sense? Okay. Okay, I'm going to move on from the flavin or the riboflavin-derived redox cofactors and touch on vitamin C. Vitamin C derived from citrus fruits, of course, and other sources. Similarly to the riboflavin-derived redox cofactors, the vitamin C can exist stably in this half-reduced or semi-quinone form after a single electron transfer. Now, the difference is that vitamin C is soluble. It's not a prosthetic group like FAD is. So vitamin C, in addition to its role that depends on its association with an enzyme, just vitamin C in a solution can have an antioxidant effect. So it can stabilize free radicals by taking an electron one at a time, and because of being somewhat stable in this semiquinone form. So one of vitamin C's roles doesn't depend on any enzymes, it's just an antioxidant all alone. In terms of its association with enzymes, it's a redox cofactor, and some of the important roles that it plays are in collagen maturation and in oxygen sensing and then in neurotransmitter synthesis. So one of the key roles that vitamin C plays is as a cofactor for dioxygenase molecules. Dioxygenases use molecular oxygen as a substrate, and they split it and add a OH group to another substrate. So two important enzymes that use vitamin C and are important in collagen synthesis are the proline hydroxylases and the lysine hydroxylases. So this is a post-translational modification of those two amino acids that occurs after translation. Okay, so it adds an OH to the proline, and then there's a similar enzyme that adds an OH to the lysine. Now, just going back, if you remember at the very beginning of the class, there was an FAD-dependent enzyme that I showed the kinetic properties of, and that was a lysine hydroxylase. That was actually the N5 lysine hydroxylase, so that was hydroxylating this nitrogen, and that's a different enzyme than the one for collagen maturation, which is hydroxylating a carbon. Okay, so this, and you'll notice alpha-ketoglutarate is a substrate, and the decarboxylated succinate is a product. So alpha-ketoglutarate and succinate are TCA cycle intermediates. So there's some overlap between the TCA cycle and this family of alpha-glutarate dependent dioxygenases, of which the lysyl hydroxylase and the prolyl hydroxylase are members. Okay, so the function with collagen of that hydroxylation of the prolines and the lysines is to add additional electronegativity and additional hydrogen bonding capacity to the amino acids that make up collagen. So collagen is, I think it's by far the most abundant protein in our body, and it's like all of the extracellular matrix and bone and tendon and what have you in our body is made up of collagen. So the collagen peptide is, it has the ability to make a really tight spiral because every third amino acid in the primary structure is glycine. And recall that glycine has the least amount of steric hindrance of all the amino acids because its side chain is a mere proton. So that's, and so it's glycine, amino acid, glycine, amino acid. These X and Ys, these are frequently lysines and prolines, not exclusively. But that hydroxylation of those lysines and prolines allows them to have additional hydrogen bonding that can allow 3 of these strands of the collagen peptide to twist together in that collagen helix. So this is a example of a tertiary structure that is a tertiary and quaternary structure that is unique to collagen. So this isn't shared with other tissues. Okay. So ascorbic acid, in terms of where we get it in the diet and the consequences of the deficiency, it's present in citric acid, in citrus fruits and in vegetables. The deficiency in ascorbic acid is directly related to its role in collagen synthesis. Okay, so that collagen is important for connective tissue, and so the manifestations of the vitamin C deficiency or scurvy are in connective tissue tissues. So you can get a loss of the integrity of extracellular matrix that can promote bleeding. For example, nosebleeds or bleeding in the gums is shown here. You can also get bleeding in capillary beds, and that can cause a petechial rash. And there's also issues with the bone and cartilage, and you can get enlargement and inflammation of the costochondral junctions, which is the junction between the bone and the non-bone part of a bone, I guess. Okay. Okay. Okay, so you would diagnose this, of course, with the clinical presentation and what the symptoms are and what the patient's diet history is, and et cetera. There are radiologic findings. If you look at those bones, there's kind of characteristic defects that you can find. Measuring vitamin C in the blood is possible, and we'll see an example of that towards the end of class. You can also do give a person a large dose of oral vitamin C and then see how much of it they urinate out afterwards. Okay, if you give them a lot of vitamin C and they're peeing it all out, the interpretation there is that they have plenty of vitamin C. Okay, if you give them a large dose of vitamin C and they don't pee it all out, that means their body is starved of vitamin C. Okay, so, okay, questions on vitamin C? Yes.
Not about vitamin C specifically, but before you had a couple arrows where like the name of the enzyme was on top and then the cofactor was below. Is that kind of a standardized, like what we see, if we see that again, the enzyme's on top and then we could consider the lower one a cofactor?
That's a common convention. Just consider those cofactors though, be savvy. Those cofactors are substrates and products of the reaction as well, so they're not like unimportant, I guess. Well, yeah, good question. Okay, metal atoms are common accessories or cofactors for redox enzymes, and the useful redox metals are metals that will exist stably in more than one oxidation state. Okay, so they can be oxidized or reduced and they're stable in either form. So that makes them good accessories for partnering in redox reactions. I'm going to just talk a little bit about iron. And so iron is important in hemoglobin and in myoglobin. There are also iron sulfur clusters that are important in redox reactions. So complex 1 of the electron transport chain, I think I may have mentioned that. That's the main job of that is to oxidize NADH to NAD+. And that uses iron in the form of iron sulfur clusters as a quantum wire, if you will, that is going to transfer electrons from the NADH, goes through flavin mononucleotide, and then it reduces this iron, and then this iron gets oxidized, and then it reduces this iron, then reduces this iron, reduces this iron, so on and so forth, until it ultimately, the electrons travel through that quantum wire down to coenzyme Q. Coenzyme Q is reduced and then diffuses away and transfers the electrons to complex 3 in the electron transport chain. Yes?
Is there a reason to have all those irons and not just have one iron transfer electrons to that iron and then just have it be done? Is it just more steps that regulation can happen?
I think evolution doesn't always
Act efficiently?
Yes, right. And it can be argued that, less efficiency is favored, right? So yeah, I don't have an answer to that. It's kind of the way things worked out. Other questions? Okay. All right, so let's move on to another family of enzymes, or another family of cofactors, and these are cofactors that participate in activation transfer reactions. What we mean by this is transferring a carbon substrate from one position into another position. Okay, so moving carbons around. Some examples, we'll talk mostly about carbons, but kinases would be in this category as well, and they're transferring phosphates around. So transferring things besides electrons around, let's say. Okay, so the cofactors that we'll talk about, lipoate, coenzyme A, biotin, pyridoxal phosphate, and cobalamin. Cobalamin, we'll talk about this quite a bit in a separate lecture, so I'm just going to touch on that. So lipoate and coenzyme A and thiamine pyrophosphate should be on here too. That's an omission. Those three, plus the redox cofactors NADH and FAD, are all participating in one enzyme complex that is pretty important for fuel metabolism, and that's the pyruvate dehydrogenase complex. So I'm going to talk about the pyruvate dehydrogenase complex for a little bit, both touch on its importance in its role in metabolism, but mainly it's a model for different cofactors and what they do. Okay, so pyruvate dehydrogenase, where it fits in metabolism is it links glycolysis with the tricarboxylic acid cycle. Okay, so glycolysis is taking the 6-carbon glucose and making it into the 3-carbon pyruvate. Pyruvate dehydrogenase then takes 1 carbon off of pyruvate as carbon dioxide, and then it takes the other 2 carbons and attaches them to coenzyme A. Okay, so acetyl-CoA, this is the product of the reaction. While it's doing this reaction, pyruvate dehydrogenase also has a redox function, and it's transferring electrons from the pyruvate substrate onto NAD+ to make NADH. Okay, so it has both redox and transfer functions. So the whole reaction shown here, NAD+ and pyruvate, coenzyme A, if it's just free coenzyme A, it's frequently written as coenzyme A, SH, reflecting the reactive atom as a sulfur at the end of the coenzyme A. Then the products are the reduced NADH, acetyl-CoA, and carbon dioxide. And again, where this fits in metabolism, the pyruvates are derived mainly from dietary carbohydrates. We can get them from other places, but mainly from there. The NADH and more NADH that can be generated through the conversion of acetyl-CoA to carbon dioxide, that then can generate ATP. Okay, so let's look at this enzyme and all the cofactors that are involved. Okay, so it has 3 different transfer cofactors, thiamine, lipoate, and coenzyme A, and 2 redox cofactors, FAD and NAD. Okay, so each one of those is associated with a different function of the catalytic cycle of the enzyme. So the first one that we'll talk about is thiamine pyrophosphate. Thiamine pyrophosphate is important for decarboxylation reactions. Okay, so the thiamine pyrophosphate shown here, derived from vitamin B1. This is bound to the pyruvate dehydrogenase as a prosthetic group. The reactive carbon is shown here, so the reactive carbon will bond to its substrate, pyruvate, and in doing so it makes an electrophilic attack and then releases carbon dioxide. So this reactive atom is binding to this carbon in pyruvate, the ketone carbon in pyruvate, and then this carbon goes off as carbon dioxide. So after the thiamine pyrophosphate step in the PDH reaction, you get the 2 carbons from pyruvate are covalently bonded to the TPP substrate. Clear? Okay. So let's talk about where TPP comes from for a moment. So vitamin B1 is the source of thiamine in our body. When that gets taken up, that is phosphorylated within cells to make the thiamine pyrophosphate. That thiamine pyrophosphate then, that's the active cofactor that can bind to enzymes like PDH. So there's a free pool of thiamine pyrophosphate, and then there's an enzyme bound portion as well. We can also remove the phosphates through a phosphatase, but just don't worry so much about that. I'm mentioning this SLC19A transporter. This is the way that thiamine can enter into the cells. There are inborn errors of metabolism resulting from a loss of function mutation in that transporter that can recapitulate the symptoms of thiamine deficiency. The symptoms of thiamine deficiency are referred to as beriberi, and in the Togolese language, beriberi means I can't, and that's kind of the hallmark phenotype of that weakness and malaise and just generally not having any energy as one of the hallmark features of beriberi. You can also have peripheral neuropathy, or like tingling and numbness in the fingers and toes. There is a wet and dry version, and the wet version, that is associated with a cardiac myopathy, and that can definitely cause death. There's a condition that's often lumped together as Wernicke-Korsakoff, but there are two subtly different changes in cognition and mental capacity that are also associated with beriberi. And then this thiamine-responsive megaloblastic anemia, that's that deficiency in that thiamine transporter that recapitulates the dietary deficiency. Okay, so let's move on from thiamine to lipoate. Okay, so in our catalytic cycle with the pyruvate dehydrogenase, we've taken pyruvate and decarboxylated it. We remove carbon dioxide, and then 2 carbons are bound to the thiamine pyrophosphate cofactor. Okay. The next part of the catalytic cycle is going to transfer those 2 carbons from the thiamine pyrophosphate to lipoate. Lipoate is, so this is the only cofactor that we'll discuss that doesn't have a vitamin precursor. So lipoate, we can make this from sulfate and acetyl-CoA and amino acids. So lipoic acid, it has 2 sulfur atoms. Similarly to glutathione, those sulfurs can be oxidized, as shown here, or reduced by the addition of electrons. So in our reaction, this molecule, hydroxyethyl TPP, this is those 2 carbons from, ultimately from pyruvate, bound to thiamine pyrophosphate. So the hydroxyethyl part, the 2 carbons, those are going to get bonded to this sulfur. They're also going to be carrying some electrons with them, so they're going to be reducing the other sulfur. Okay, so then the TPP, not shown here, but that would be a leaving group. So it's a handoff of those 2 carbons from thiamine pyrophosphate to lipoic acid. And as I mentioned, those sulfurs, these sulfurs are kind of a redox component of our electron, or of our activation transfer enzymes. Okay, so lipoate doesn't have a vitamin precursor. There are conditions associated with an inability to synthesize it. So those are rare inborn errors of metabolism that are associated with epilepsy that would, start early in life, but those are extremely rare. So a dietary deficiency of lipoic acid is not described. Okay, so in our catalytic cycle now, we've got 2 carbons from pyruvate. 1 carbon goes away as carbon dioxide. The other 2 carbons get bonded to thiamine pyrophosphate, then to lipoate, and then as the final destination of those 2 carbons, they bond to the sulfur atom on coenzyme A. So coenzyme A, so this is the sulfur atom, this binds the acetate substrate, and then the rest of the molecule is shown here, and we have this common nucleotide structure that acts as a common recognition handle for the molecule. So that adenosine nucleotide portion of it, of course, we can synthesize that just fine. But that intermediate section between the nucleotide and the reactive sulfur, this is the part that we can't synthesize and that we need to have a vitamin precursor for. The vitamin precursor is pantothenic acid, or vitamin B5, and there's a shampoo called Pantene, if you remember that. So that's it. Vitamin B5 is supposed to be good for your hair. I'm not sure how, but if that helps you to remember it. Pantothenic acid is the precursor for coenzyme A. And coenzyme A, or excuse me, pantothenic acid is very widely distributed in the diet. So there's not a natural deficiency of it that's described. If you have an experimental model where you can make a vitamin B5, completely devoid diet, in birds that results in depigmentation and defeatherization. So it's kind of it's evidence that it's good for hair, I guess. But anyways, it's not something that you're going to see a deficiency of in your patients. Okay, so let's review our pyruvate dehydrogenase reaction. We've got our 3-carbon pyruvate up here. That gets 1 carbon goes away as carbon dioxide. The other 2 carbons go to thiamine pyrophosphate, then to lipoate, and then to coenzyme A to make acetyl-CoA. Okay, and then acetyl-CoA, this can be a substrate for TCA cycle or any other purpose. Okay, so this cartoon here, I think that it's a little crude compared to modern art, but I think it's a very useful depiction of the pyruvate dehydrogenase catalytic cycle. So first off, you'll note there's different protein subunits, right? So this is a multimeric enzyme. Different subunits of the enzyme bind different cofactors and are responsible for different catalytic functions. Okay. All right, so I'm going to try to just go through this pretty quickly. Okay, so here is our pyruvate. As the first reaction, it's decarboxylated, and the 2 carbons go to thiamine pyrophosphate. Then the 2 carbons go to lipoate, and then there's actually multiple lipoates. So it's going from this lipoate to this lipoate, and then to coenzyme A. Okay, so coenzyme A then leaves, and other stuff happens to it. Okay. Now you'll notice when coenzyme A leaves, one of the lipoates is in its reduced form. You see that? Okay, so the rest of the reaction is a redox reaction. You get transfer of electrons from this lipoate to this lipoate. The electrons then transfer to FAD to reduce it. Then the electrons are transferred to NAD+ to make NADH, and then the enzyme is restored. Okay, clear? Okay, so this is showing the 3D reconstruction of the enzyme. So this is it's a big deal within the cell. A lot of different, repeats of the same subunit. So I just ask you to, stew over this for a few minutes on your own, and if you have questions about it, come and visit with me, and we can go through it. Okay, so I'm going to talk about some other activation transfer cofactors that are not associated with pyruvate dehydrogenase to conclude the class. Okay, biotin. Biotin is a cofactor that is covalently bound to lysines within proteins, and the catalytic function that it participates in is the opposite of thiamine pyrophosphate. So thiamine pyrophosphate participates in decarboxylation reactions, and biotin participates in carboxylation reactions. Carboxylation being taking carbon dioxide and some organic substrate and adding that carbon in a new carbon-carbon bond. Okay? So there's only 4 enzymes that biotin participates in within our body. So acetyl-CoA carboxylase, pyruvate carboxylase, propionyl-CoA carboxylase, and methylcrotonil-CoA carboxylase. We're going to talk about all of these, except I think this one, I'll maybe touch on that a little bit. But so don't worry so much about what those enzymes do. I've got their broad function there. But just remember that biotin is associated with the carboxylase. Okay, so an example, pyruvate carboxylase shown here, which is important for gluconeogenesis. That's taking carbon dioxide, or shown here bicarbonate, and making a new carbon-carbon bond with a substrate to make the dicarboxylic acid oxaloacetate. Okay, so the other three enzymes that biotin participates in, it's the same essential deal. It's taking carbon dioxide and making a new carbon-carbon bond. Okay, another kind of crude diagram here that has all the hallmarks of 1950s U.S. Department of Agriculture, but it's useful, and I haven't found anything better. So the main principle here is that biotin, when we eat it, remember, biotin functions in our body associated with proteins, right? Covalently bound to proteins. So some of the biotin that we eat in our diet is from protein that we eat. Okay, so you can get protein-bound biotin. As the protein is digested, then that's going to release free biotin, and that becomes available for our apocarboxylases to be converted into holocarboxylases, right? And then when these holocarboxylases, when these get digested away, when their life is done, then that releases biotin. Okay, so that's not a huge deal, I guess, but just wanted to illustrate that. Okay, so like pantothenic acid, biotin is widely distributed in food, and a biotin deficiency is rare. You can describe it experimentally, and it's got alopecia and dermatitis are some symptoms if you have laboratory animals that you can eliminate the biotin from. Where you would see this in people is in people who eat a lot of raw eggs, which was, well, does anybody remember that from the movie Rocky? Okay, so it was something that bodybuilders used to do, I think, before all those protein powders and stuff came on the market. And there's a protein within eggs called avidin. And avidin, if you cook the egg and denature it, you destroy its binding activity, but the native avidin will bind up biotin in the diet and make it not able to be digested. So that would be an example of a functional biotin deficiency, right? You're eating enough biotin, but you can't absorb it and use it. Yes?
Is it true to say that cofactors like these vitamins, is biotin a vitamin?
Yes, we can't take, like, acetate or glucose or something and synthesize biotin ourselves.
So are they kind of like enzymes in the way that like they're used to make reactions happen, but they're not used up as long as the redox reaction is reversible?
The redox, yes, so the cofactors will generally have to be restored by some other reaction. Now, in the case, not everyone does. In the case of coenzyme A, for example, there's energy that comes from outside the cofactor to make the high energy bond between the substrate and the sulfur, and then when that bond is broken, there doesn't have to be any other reaction to make the to regenerate coenzyme A. So yeah, cofactors are essential for catalysis. I think I'll just leave it at that. They're essential components of enzymes, and the enzyme can't really function without them. But they're non-protein components of enzymes.
But they don't get like degraded through the reaction, is what I'm kind of getting at.
Oh, no. If they do get modified, there's another reaction that will restore them. Other questions? Okay. The next one that I'm going to talk about is pyridoxal phosphate. This is derived from vitamin B6. And pyridoxal phosphate This is a cofactor for hundreds, many enzymes that will use an amino acid or something that looks like an amino acid as a substrate. So this reactive group, this reactive carbon on the pyridoxal phosphate, this has a particular affinity for binding onto that nitrogen on the peptide backbone. So for what, whatever three-dimensional shape that makes, it's perfect for binding into the peptide backbone of an amino acid. So once the cofactor makes this bond, then some other portion of the enzyme in the active site can modify that amino acid by being either a transamination or a decarboxylation or what have you. So the pyridoxal phosphate containing enzymes grab onto the amino acid, and then the enzyme does whatever it's going to do with it. Okay. So pyridoxal phosphate, vitamin B6, think amino acid metabolism. Okay, so pyridoxal phosphate is shown here, and we can get it through, these are all considered vitamin B6, or they're all considered pyridoxines. And we can make our pyridoxal phosphate through any of these substrates. So if we have pyridoxal, the aldehyde, this can be phosphorylated to pyridoxal phosphate. The pyridoxine, this can be, again, phosphorylated and then oxidized down to pyridoxal phosphate. Likewise, the pyridoxamine, this can be kinased, and then an amine transferase can generate the pyridoxal phosphate. So this is the active cofactor form. These are the vitamin forms that we can eat it in, and then these two are intermediates for how we get there. So let's see, fortified cereals, like many of these vitamins, you can get it in a cereal or a multivitamin pill. Meat, and particularly pork, is very rich in vitamin B6, but there are some vegetarian sources as well. Okay, so the pyridoxal phosphate or vitamin B6 deficiency manifests itself as neurological conditions, and it's more severe if it's an infant presentation than if it's an adult. If it's an adult, it's going to present as peripheral neuropathy, like weakness and numbness and tingliness in the fingers and toes. If it's an infant, it's more severe, and that can have the neuropathy symptoms as well as seizures and more significant abnormalities. Remember that we can get niacin from tryptophan, right? But in order to get niacin from tryptophan, we need pyridoxal phosphate. And that's shown here in this, don't commit this to memory. This is the kynureninase pathway, and it's lengthy, but there's one step that requires pyridoxal phosphate. And if you look at the structure, we've got an amino acid structure, so it's not surprising that one of the enzymes is going to require it. So the bottom line here is that if you have a vitamin B6 deficiency and you're making most of your niacin from tryptophan, the vitamin B6 deficiency can also recapitulate the symptoms of a niacin deficiency. Okay, so the diagnosis of a deficiency, you look for the symptoms, of course. You would look for an enzyme that requires pyridoxal phosphate, like a transaminase. You can measure the PLP in the blood, or you can just give back vitamin B6 and see if the symptoms resolve. Okay. Oh, and vitamin B6, now many of the water-soluble vitamins are not toxic or minimally toxic, but vitamin B6 is one that has a described maximum tolerated dose. The symptoms of the overdose are kind of recapitulating the symptoms of the lack of the enzyme. Okay, so kind of interesting. Okay, there are two inborn errors of metabolism associated with pyridoxal phosphate metabolism. Pyridoxine oxidase deficiency is a deficiency in the pyridoxine oxidase, and you can't make pyridoxal phosphate from the pyridoxine in that case. The symptoms are essentially the same as a vitamin B6 deficiency. They would manifest in infancy because it's an inborn error of metabolism. Okay. Now, if you have an infant with a vitamin or with the symptoms of a vitamin B6 deficiency, you can try to give the child one of these other forms of vitamin B6 to see if it resolves. Okay. There's another inborn error of metabolism that is associated with vitamin B6 deficiency. This is antiquitin deficiency, and this is a case where the lack of antiquitin makes this other compound called pyric acid, and that binds with the pyridoxal phosphate and makes it not available for the enzyme. So it's a different enzyme involved in a different product, a different pathway, but it locks up this product. Okay, so if you have this condition, you could give the kid pyridoxine, like huge amounts of pyridoxine, and the kid could still make pyridoxal phosphates through this pathway. And if you give them enough of it, it's going to have enough of this available. Is that clear? If you have this condition and you give the kid pyridoxine, it's not going to get better. Is that clear? Okay, so one of the ways, if you suspect the kid has one or the other of these, you can just give the kid pyridoxine, and if he gets better, he doesn't have this, he's got the other thing. Okay, so that's a kind of old school way to distinguish between the two. Now you would probably do sequence the genes and look more directly. Okay, this is that other pathway where antiquitin is present, and don't worry so much about that. Okay, vitamin B12 is the final activation transfer cofactor that I'd like to discuss. Vitamin B12 is present in our diet from animal sources, so vegans can be susceptible to vitamin B12 deficiency if they don't have some other source of it. So the structure is shown here. So there's a cobalt atom in the center. Then we have this 4-ring structure. Then below the ring, kind of, into the screen is an adenosine nucleotide. And then over the cobalt atom is either a methyl group or another nucleotide. So in our body, this is either deoxyadenyl cobalamin if this is a nucleotide, or it's methylcobalamin if this is just a methyl group. Okay, so vitamin B12, biochemically it's pretty easy. There's only 2 reactions that it participates in. And both of them involve the transfer of a methyl group from one substrate to another. In the synthesis of methionine, vitamin B12, or methylcobalamin, this is the donor that provides the terminal methyl group for methionine. In the biosynthesis of methionine, its use in making a protein. But methionine is also an important intermediate in the synthesis of a lot of other things. So methionine can be converted into S-adenosyl methionine, and then S-adenosyl methionine, that's like a universal methyl donor, or not universal, but it's a methyl donor for many, many reactions that would involve methylation, like conversion of norepinephrine to epinephrine, for example. Okay, so let's so that's one methyl transfer activity, and that's using the methylcobalamin. Adenosyl cobalamin, this is important for the catabolism of branched chain amino acids and branched chain fatty acids. And the catalytic activity is to resolve a branch. Okay, so we have here carbon, carbon, carbon. So this is the linear part of the molecule. And then we have a branch of this methyl group over here. So adenosylcobalamin is a cofactor for the methylmalonyl-CoA mutase, and the enzymatic function is to resolve that branch. So it's moving that methyl group from here over to here. So now we have 4 carbons that are all lined up. That then can be a substrate for oxidation in the TCA cycle. So this is, normally the fuel metabolism pathways don't like branched molecules, they like linear molecules. So the methylmalonyl-CoA mutase, this is a way that we can resolve those branches and get useful energy from branched chain products. Okay, so cobalamin deficiency, the most common forms of cobalamin deficiency, at least in the US, are caused by a failure to take it up in the diet. So autoimmune destruction of the parietal cells that are important for making vitamin B12 bioavailable, or other aspects of cobalamin metabolism that make it unavailable. So people, again, who are vegans can be susceptible to vitamin B12 deficiency, but generally vegans kind of, are aware of what they eat and know about this issue, so it's not so common. The key hallmark symptoms, I guess, are anemia, and it's a megaloblastic anemia because of a failure to make nucleotides, because that methionine cycle gets all messed up. Okay. And when you have an anemia, you get weakness and fatigue and things that are associated with it. So megaloblastic anemia, remember, that is also associated with folic acid deficiency. But remember, vitamin B12 has this other role in synthesizing neurotransmitters, for example. So we have additional neurological symptoms associated with a cobalamin deficiency. Okay, that's it. Okay, so I want to get to the case. I'm going to say a couple of things about this slide. When we're all done with fuel metabolism, review this slide, and it's going to make a little bit more sense. Essentially, this is showing, okay, the pathway from carbohydrate to pyruvate, what cofactors do the enzymes require? Pyruvate to acetyl-CoA, this is that pyruvate dehydrogenase reaction, so this is all the cofactors. Okay, so I'm not going to go through this because we haven't gone through those pathways yet. I've got a couple tables here. If you and your study buddies kind of fill this table out while you're studying, you'll never have to look at the table again. You'll know it, okay? All right, clinical case. So this is, I wanted to make sure we have enough time for this. Okay, so one student recognized this as scurvy, right? Does scurvy make sense now? Okay, so the question is, what would you do? So what would you do with a child if you suspected scurvy? And I think that the two most common answers were ask about the diet and measure water-soluble vitamins in the blood. Okay, so let's see how this case really unfolded. Two-and-a-half-year-old kid, bone marrow biopsy, muscle biopsy, MRI, was it? Yeah, multiple MRIs. Then they decide they're not going to ask what the kid eats, they're just going to measure the vitamins. So, good. Okay, vitamin C was 0.1 milligram per deciliter, and the reference range should be 6 times or more of that. So vitamin C deficiency, right? Okay. Let's do clicker question. Okay, so I want to, just looking at the clock here, I'm going to leave this just for your own review. But I think this is an example of how you could make a two-step question here. Okay, so after they found out that the child was vitamin C deficient, they reviewed those x-rays. So I guess they were just looking for fractures in the x-ray and they didn't look closely, but they re-examined them and there's this little kind of shadowy area right at the plate between where the collagen is converted into bone. Okay, so they missed that first and then they noticed it. Okay, and then they decided to ask about the dietary history and what was this kid eating? 1.4 liters of chocolate milk and 4-gram crackers per day. Okay, so supplemental ascorbic acid was started, and the dose, was a high dose, and that the symptoms resolved. Okay, this occurred in the United States, and I think this came from suburban Detroit or something like that. So not, people should know better. And frankly, the physician should know better, right? I mean, how much does it cost to ask a parent what their kid eats? Nothing. What does a bone marrow biopsy and flow cytometry cost, and multiple MRIs, muscle biopsy? Okay, so bottom line, diet is important, and there are easy questions that you can ask your patients that can help identify conditions like this, or help to exclude conditions like this from your differential. Okay, good. All right, so this is a kind of a cool case. You can read it there in New England Journal if you like. There's a bunch of other clicker questions. I'm going to post these, and I'll just leave them for your review. If you have questions about them, ask me. Okay. All right, sorry for going over, but thank you and enjoy your weekend.
The last one you'll ever have.
I know, so I feel like I want to see. How did you cite this article with it not applying? A better article. The first one just reinforces and voices. Oh wow! About the experience, or I was wondering, I was around right here. No, when I looked around, we're all
That's 20. Oh,
yeah, like kind of feels the very I've read it. Go for it. Yeah, he was going on over there. I do like that this lecture included those topics. I was like, okay, context. But I was like, at least the second half of the lecture again. Honestly, I was like, I think about when add an extra thing to what just happened. I feel like I heard you know No, it's funny you make that descriptor because the first week I was oh my god, it wasn't quite. I was it's but I understood sort of a garden hose on the door. It's
You probably have a deficiency. No, I'm not like necessarily, they just have a risk of them, but. Oh, yeah.
I am.
I'm not. So I am.
No, you're good.
I'm gonna be doing.
I hope it goes.
Yeah, anywho, we should study.